This next chapter, chapter 4, talks about the cell. It's called A Tour of the Cell. It talks about the organelles within the cell and kind of what their function is and how it relates to the structure of cells. So this overview is really just shows you two different types of cells. One would be a nerve cell here. You know, they're long and then they connect one thing to another, and they're able to send messages. And this would be similar in that it's a nerve cell, but now it's connected to skeletal muscle. This is skeletal muscle, and skeletal muscles don't contract, if there's not a connection to a nerve. So anyway, these are just pictures of cells, specifically different nerve type cells, which come to be the simplest units of living matter, meaning all life is made up of cells. And when you break that living thing down to its simplest unit, that would be a cell. And cells descend from earlier cells. What that means is that cells, it takes a cell to reproduce a cell, or you get a cell from a cell. Cells just don't come out of nowhere. So cells are inherited or passed on to the next generation, the next life form. 4.1 talks about types of tools that are used to see cells, and the microscope is very old and has been modified and upgraded and improved over time. But this is still one of the best techniques to use and best tools to use to study cells. Cells range from very small units. Cells are measured in something called a micrometer or a micron. This is 1/1000th of a millimeter, so very, very small. And we need tools in order to see things that are that small. But they range from about 1 to 100 microns or micrometers. We use something called a light microscope, which this is an example of one where you have a light source that shines on a mirror, and then that image is projected ultimately to the viewer's eye that's looking through the eyepiece. So with a light microscope, this visible light passes through the specimen and then through the lenses ultimately to the eye. Specimens need to be very thin so that that light is able to shine through. And we'll be doing a lab fairly soon which uses that technique of setting up a slide, putting it on a microscope, and then looking through the microscope in order to see the different structures within that cell or that tissue, see the cells within that tissue. With these lenses in the microscope, they bend the light, which is also called refraction, and that is then going to magnify the image. So we can see here, here, that image is, and here also. These lenses magnify that image by bending it so that it looks bigger to our eyes. With microscopes then, we see really small things. So we've got this organism up here. Things are getting smaller as we go down. You know, a nanometer is 100,000 times smaller than a millimeter. We have 1 micron, which is 1,000 times smaller, and then we have this thing called a millimeter. So an egg, for example, like a chicken egg, if we're going to about here, you know, we've got a meter, and then we go down to, you know, say a millimeter, which is 1,000 times smaller. That then, if we 1,000 times decrease the size again, that's where we get down to this micron or micrometer. With cells then, you know, here would be a bacteria cell. This is going to be most plant animal cells are within this 10 to 1,000, or excuse me, 10 to 100 micrometer range. So we need this light microscope, which is abbreviated by the LM. It kind of shows you the range of that. We'll also talk about things called electron microscopes, which can see much smaller particles, including viruses, which are even smaller. But with magnification, the lenses, etc., are used to increase that object size to, you know, from 1 centimeter basically to real size. So object's image size is increased. Resolution is the ability to see clearly, so the better the microscope, the more clarity, the better detail you can see. And that's kind of identified as being the distance between two distinguishable points. So the better the microscope, the better you're able to resolve two very close points. The closer they are, the better able you are to resolve the difference between those two, the better the microscope. And so more expensive scopes, you can have better resolution and have more clarity with things that are much smaller. Contrast is the ability to see the differences. A lot of times we'll use a stain to contrast the varying parts within a cell. And again, when we do the cell lab coming up, you'll use stains in order to see parts of cells and cells themselves a little bit more clearly, because you can distinguish, and there's visible contrast between different parts. Light microscopes then magnify things about 1,000 times. We'll do a microscope lab coming up where you're going to use the light microscopes, and there are actually 4 different lenses. We'll use 3 of the 4 more typically. There's a 4x, meaning 4 times what the naked eye can see. That's called the scanning lens. There's a 10x, which is 10 times what the naked eye can see. That's a low power. There's a 40x, which is the high power, 40 times what the naked eye can see. There's actually a 100x, which is an oil immersion lens, magnifying 100 times what the eye can see. We don't use the oil immersion as much, but given these different types of lenses, then we can magnify things to much greater detail than what the eye can see. We also use these staining techniques in order to see things a little bit more clearly and have better contrast. The electron microscopes are a different type of microscope using electrons, not light. And with these, we can see organelles or structures that are even smaller. We can go, about 100 times again what we can see with the light microscope, so down into the nanometer range. There are 2 types of electron microscopes. There's a scanning. This thing scans over the surface and you end up with a 3D image. The cilia here would be taken with a scanning microscope where you can see the actual structure of the external surface of something. So it's pretty cool. They also use staining techniques, but this would be where the electron beam provides this 3D surface image of something. So it's called a scanning electron microscope. Transmission is one that transmits the electrons through the specimen, so you're looking more internally. This is a transmission where you're seeing a cross-section of those cilia. You're seeing kind of that internal structure of those cilia. So these need to be very thin, and you have to pass the electron beam through them, and so we transmit that beam through that specimen. So these are 2 different types of electron microscopes, both having much greater magnification ability compared to the light microscope, but as is similar to the light microscope, these magnify things so that we can see things in greater detail. With light microscopy, we're able now to use a couple of different techniques. One is fluorescence marking, where you can actually take it's like a radioactive material that you can cause this fluorescence so that you can see things a little bit more clearly and brightly, and you have better contrast with them. Confocal microscopy sharpens the image of tissues and cells like that. So here we have, different contrast ability, so we can see things a little bit differently and better. And also, we have improved resolution up to about 10 to 20 micrometers with these different techniques with either the fluorescent or the confocal microscopy. Self-fractionation is where you're able to separate components, and this is basically using a centrifuge which spins out things according to their weight and according to their relative size, and so things kind of fraction off at different times depending upon how large they are and how much they weigh. And this scientists are able then to fraction off different organelles within the cells and maybe study them a little bit more in detail in order to determine more closely what their function is. Biochemistry and cytology have these abilities and these techniques And what they're trying to do is correlate the cell function with the structure, similar to like a protein. The look of the protein relates to what it does. With cell organelles, the look of that organelle oftentimes is correlated with its function. So with cell fractionation, we can fraction off using this technique, fraction off these cell organelles, and then look at those organelles in more detail to figure out what their function is according to their structure. 4.2 goes into the different parts of cells, the organelles contained within. But in general, there are two basic types of cells. One is a eukaryotic cell. There is also a prokaryotic cell. That prefix relates to the type of cell it is. A prokaryotic cell would be similar to this type of cell, which is a bacterial cell. These are very simple. They do not have a nucleus. They have DNA. They are able to reproduce. They're considered to be alive, but they don't have DNA within a nucleus. Prokaryotic cells are one of the first life forms on the planet, are very ancient. So the pro actually means before. Karyotic relates to the nucleus. So since these are very old, ancient types of life forms, they evolved before the nucleus and are very successful, but just don't have that wrapper around the nucleus. Eukaryotic cells, eu means true, so they have a true nucleus, which you see right here and you see right here. That nucleus wraps around, contains the genetic material, the DNA, and kind of protects it from the rest of the cell. So these eukaryotic cells evolved a little bit later, and as such, they now have these membrane-bound organelles like a nucleus, which is going to protect whatever structure is within. And in the case of the nucleus, it protects the DNA. All cells have a couple of structures. They all have a plasma membrane. We talked about this a little bit with the phospholipids. We'll talk more about it in chapter 7 when we talk about kind of cells and the membrane's function, how it allows things to move across. So anyway, we call this the plasma membrane. It is really the cell membrane, which contains all the structures of that cell within it. Within the cell membrane, then, there is a semi-fluid substance called the cytosol. It's also called the cytoplasm. It's just the liquid portion of the cell from the nucleus to the cell membrane. Chromosomes are also found in all cells. Even bacteria cells have chromosomes. These are the things that carry genes. Bacterial chromosome looks different than eukaryotic organism chromosomes, but they both have genes within the chromosome, which can then be inherited and is able to be passed on to future generations. All cells also have ribosomes, the ability to make proteins. So even bacteria cells have ribosomes, along with eukaryotic cells, which also have ribosomes. And that ribosome's job is to build protein. Prokaryotic cells are characterized then by having no nucleus. They are very simple structures. They have a region within the cytosol, which is called the nucleoid region. This is where the DNA is contained. Right here you can see it's this purple structure. And in looking at that, there is no nucleus around that, but they do, again, contain chromosomes. It's usually a circular, singular bacterial chromosome. But in that chromosome is the genetic material containing the genes, coding for all the different traits of that bacteria cell, which can then be passed on to future bacteria cells. There are no membrane-bound organelles. They do have a cytoplasm, which again is also called the cytosol. They do have ribosomes. And with bacteria cells, or these prokaryotic cells, recently scientists have divided them into two domains, bacteria and archaea. Both are bacterial types. The archaea are kind of the extremist bacteria that you see at like deep ocean vents, etc. You know, the hot springs. They can live in really extreme environments. We are actually more closely related to the archaea domain. The other domain, bacteria, would be those types of bacteria that live typically in an oxygenated type of environment, like E. coli, strep, staph, all those different types of bacteria that we tend to get sick from if they end up in places they shouldn't. But the bacteria domain is a little more has a more distant common ancestor. We have a more recent common ancestor with the archaea and are a little bit more closely genetically related to the archaea bacteria. Eukaryotic cells then do have a nucleus, so the nucleus would be the blue structure here, kind of that is wrapped around with this nuclear membrane. So this would be that that membrane bound structure that we see. You know, the mitochondria would also be membrane bound. We see these membrane bound organelles within the eukaryotic cells, and that includes the nucleus. So we've got the DNA is in the nucleus. Within that nuclear envelope, there are other membrane-bound organelles like the mitochondria, the smooth endoplasmic reticulum, the rough endoplasmic reticulum. There's something called the Golgi apparatus. There are lysosomes, etc. These all can have, I should say, a membrane that binds and kind of bounds the borders of these organelles. There is cytoplasm or cytosol between the cell membrane or plasma membrane and the nucleus. And the protists, fungus, plants, and animals would be 4 types of eukaryotic cells that we have on this planet. So basically all life on this planet would be eukaryotic. The one form of life that is not would be the prokaryotic bacteria type of cell. Plasma membrane is this outer membrane, and we'll talk more in chapter 7 about how things move in and out of this membrane. It is considered to be selectively permeable, meaning it does not allow everything to cross. So certain things like gases, some nutrients, some weights are easily transportable, but then other things, there's a little bit more involved to their passage in that maybe they need a transport channel to get through, or maybe there's a hormone that needs to come along and open up a gate kind of thing. But the fact that the plasma membrane selects what can get across and what can't get across, we call it a selective barrier. It is a double layer of phospholipids. Again, we've got this hydrophobic region in the middle. This region includes the bilipid tail layer, and then we have this hydrophilic region, which includes the layer of phosphoric heads on either end. And so you have the internal environment with the polar heads associated with it, because the watery cytoplasm is attracted to the charged polar heads. You have the external region with the polar heads attracted to that, the fluid of that outer area. So again, the membrane kind of sets itself up so that the polar charged heads orientate closest to water, both on the inside and the outside of the cell, and then the lipid tails are kind of hidden in between those charged areas. The metabolic requirement of a cell kind of sets its size limit. Cells are more efficient if it's smaller, and this has to do with the fact that the surface area to volume within ratio is more efficient. You have imagine more doorways in, an easier time getting into that cell so that nutrients can cross into that cell easily, waste can cross out of that cell easily, the smaller it is. So this ratio of surface area to volume is a critical ratio, and you want a higher number on the top, basically. You want more surface area to volume, which is going to increase. If we looked at these two cells, for example, you have a 6-to-1 ratio compared to a 1.2-to-1 ratio, meaning this cell has like 6 doorways compared to 1.2 doorways. Imagine a really large school compared to a very small school. Easier to get in and out materials into that small school and waste material out of that small school because of the smaller volume compared to surface area on the outside. Organelles are compartments that function in different ways depending on the type of organelle it is. And these organelles are often participating directly with the cell membrane to transport material into or transport material out of. And there are a variety of different organelles, and for the rest of the chapter we kind of go through these different organelles. This picture shows a plant cell. Previous picture showed an animal cell. Both plant and animal cells have this membrane on kind of the interiors. Plants are a little different. They've got a cell wall external to the membrane. The membrane is still the thing that regulates what goes in and out. The wall protects and supports. But even within plant cells, we see the Golgi apparatus, we see the chloroplast, we see the mitochondria, and those are all organelles which have specific functions, and we'll kind of talk about each of those different functions. This picture just shows different organelles. Again, maybe in a cross section, this shows the mitochondria, shows the nucleus. Here we've got plant cells. You can see the nucleus along with the nucleolus. This would be yeast, so just a variety of different cell types. Sorry, the slide doesn't work in this PDF format. I apologize. But when we talk about different organelles, probably one of the main organelle, and not the most important, but is an important organelle in that it is the nucleus. The nucleus is the thing that holds the genetic instructions. So the DNA, or the genes contained within the DNA, is held within the nucleus. So that would be this organelle, this membrane-bound structure, this nuclear envelope that contains the DNA within. Ribosomes are also important in that they are kind of tied to the DNA, in that the DNA codes for the protein, what the protein is going to look like. The ribosome is the structure that actually builds the protein. So ribosomes build proteins with information from DNA. And this just shows you how closely those are associated. We've got DNA, and then right close to the DNA would be this rough endoplasmic reticulum, which is really endoplasmic reticulum that contains ribosomes on it. So directions are very close in the DNA, in the nucleus, and that information moves also quickly to the ribosome associated with the nucleus, so that protein can be built according to its instructions. The nucleus then is the thing that contains the genes, and you can see this pretty easily. We'll do a cell lab in a little while where you're going to look at cheek cells and onion cells, and you'll be able to see the nucleus. It's a large structure and can be easily viewed. The envelope is the thing that wraps around the nucleus and encloses it. It too is a double membrane. It is a lipid bilayer exactly similar to the cell membrane, but now it's within the cell itself around the nucleus. So this is a thing that kind of regulates what can leave and enter. DNA cannot leave, but a molecule called mRNA, messenger RNA, can leave the nucleus. So certain things do exit through nuclear pores, which you can see on the surface kind of the envelope. And these pores are the thing that regulates what can enter and exit. Chromosomes are contained within the nucleus. Chromosomes are structures. They are hereditary units that contain the DNA, which is what codes for the genes. Chromatin is the material that makes up chromosomes. Chromatin, I should say, is a combination of DNA and certain types of proteins, and this DNA and protein combination makes up the chromosome, and we call that DNA protein combination chromatin. With the help of the proteins especially, DNA is able to condense, and when we get to mitosis and meiosis, it is critical that the long thready strings of DNA condense right before cell division so that chromosomes can be moved around cells so that some of the copied DNA can move into new cells. And we'll talk more about that in later chapters. But these proteins allow the DNA to condense in a way that makes them shorter than the normal long thready look to DNA as it normally is during the life cycle of a cell. The nucleolus is also within the nucleus, and this is actually where ribosomes are produced. So it is the site of ribosomal synthesis, and in this picture you can see the darker nucleolus within the nucleus, which is where ribosomes are built. Ribosomes then are protein factories. Those are structures that actually build proteins. Ribosomes contain both RNA and protein, rRNA, which is ribosomal RNA, and proteins. With protein synthesis, there are several different types of ribosomes. We have some that are free in the cytoplasm. We also have some that are on the endoplasmic reticulum or even the nuclear envelope. Both locations contain ribosomes that actually do the same thing. They're really just building protein. So the function of ribosomes is to build protein, but we find ribosomes in several areas. One would be free in the cytoplasm, and the other would be either on the endoplasmic reticulum or on the nuclear envelope. These are referred to as bound ribosomes, but they're similar in either location in that all ribosomes really are there to build protein.